Nucleic acids are macromolecules that serve the function of carrying genetic information in the form of DNA or RNA. A nucleic acid consists of many nucleotides in a long chain. A DNA nucleotide has three main sections: the phosphate group, the deoxyribose sugar, and the nitrogenous base. Phosphate groups are connected to the 5' carbon of the deoxyribose sugar and forms part of the phosphate sugar backbone. Covalent bonds, called phosphodiester bonds, form between nucleotides. The nitrogenous bases connect to the 3' carbon of the deoxyribose sugar. There are 4 types of nitrogenous bases: adenine, guanine, thymine, and cytosine. In DNA, there are complementary pairs. Adenine pairs with thymine, and guanine pairs with cytosine. These base pairs are connected by hydrogen bonds, which connect a base from one strand to a complementary base in the other strand. Because complementary pairs are bonded together, the DNA strands run anti-parallel to each other, meaning they run in opposite directions. Before DNA replication can actually occur, the original double-stranded helix must first unzip and form a replication fork. DNA helicase breaks the hydrogen bonds between base pairs in order to form the fork, and the separated strands run in 2 different directions. While the strands are being separated, another enzyme known as topoisomerase acts at a region above the replication fork to relieve the stress caused from the helix and stabilize the molecule. The leading strand is oriented in the 3' to 5' direction, indicating that DNA replication can occur in a simple fashion. However, the other strand, known as the lagging strand, runs in the opposite direction, which creates a more difficult process of replication. The process of replication is semi-conservative, meaning 2 daughter copies are produced from each original strand. Each daughter copy conserves the information from one strand, which is half of the original DNA molecule. Next, an enzyme known as DNA polymerase III creates a new complementary strand of DNA. The strand must first contain RNA primers, which are short stretches of RNA nucleotides. They are necessary as DNA polymerase III cannot lay down the first nucleotide of a strand on its own and relies on a primer to start the new strand. RNA primers are catalyzed by an enzyme called primase, which is active at the replication fork. After the primers are synthesized, polymerase III can create the new strand. By adding free nucleotides that are complementary to the template strand in a process called elongation, the leading strand is able to elongate continuously. However, the lagging strand must elongate in segments called Okazaki fragments. The replication of a lagging strand requires multiple primers and results in many small sections of DNA rather than one continuous strand. Afterward, another enzyme known as DNA ligase joins together all the Okazaki fragments into a continuous strand like the one formed in the leading strand. During this whole process, proofreading, a process where errors in DNA replication are corrected, is carried out by an enzyme called DNA polymerase I.